Hi friends, when we born we were so small, but now our size has increased. How this became possible? We know all living organisms are made up of cells. These cells undergo cell division and this causes all living organisms to grow. We all know if we got any wound in our body, it will heal after some time. The new cells formed from cell division helps in wound healing. In the case of unicellular organisms like bacteria and amoeba, cell division results in the formation of new organisms, that is reproduction. Thus, cell division helps in growth, reproduction and healing of wounds, that is repair. How this cell division occurs? There is a complex mechanism behind each cell division. Let us understand the basic principle behind cell division. So, our topic for today is cell cycle, cell division and structure of chromosomes. First of all, we are going to discuss about cell cycle. What is cell cycle? Cell cycle is the ordered sequence of events that occur in a cell in preparation for cell division. That is, before cell division, the cell undergo a sequence of events for preparation of cell division. Here you can see a schematic representation of cell cycle. In this picture you can see 4 stages, that is G1, S, G2 and M. In the G1 phase, the growth of the cell takes place. Second stage, that is S, the cell copies its DNA. And in the third phase, that is G2, the cell prepare for cell division. And in the last stage, that is M or mitosis stage, the cell division takes place. The phases G1, S and G2 together form interphase. It is the time span between two cell division. Thus, we can say the cell cycle is composed of interphase followed by mitotic phase. The mitotic phase consists of mitosis and cytokinesis. Now we are moving to interphase. Interphase is the phase of cell cycle in which a typical cell spends most of its life. When we look through microscope, there will be no observable changes during interphase. As we already said, interphase include G1, S and G2 phases. So, the cell grows, replicates the DNA and prepare for mitosis. Even though under microscope, interphase shows no observable changes, interphase is the metabolic phase of the cell. In this metabolic phase, the cell obtains nutrients and metabolizes them, and it also grows, replicates its DNA and conducts other cell functions. Here is a picture of interphase. The cell contains a nucleus and there is a nucleolus inside it. We can also see chromatin fibers. So, it just look like a normal cell. After interphase, the cell cycle enters into next phase called M phase. M phase is actually the division phase. So, it is again divided into two phases, that is mitosis and cytokinesis. Mitosis is the division of nucleus. So, it is also called karyokinesis. Then cytokinesis means division of cytoplasm. In mitosis, the replicated chromosomes are separated into two nuclei. It gives rise to two genetically identical cells in which the total number of chromosomes is maintained. Mitosis is divided into four phases. They are Prophase, Metaphase, Anaphase and Telophase. Here is a picture which shows four phases of mitosis in cell cycle. Let us move on to first phase of mitosis, that is prophase. During prophase, the complex of DNA and protein contained in the nucleus, that is known as chromatin, that is thread-like structure present in the nucleus, condense into well-defined chromosomes. Then we can see replicated chromosome. It have an X shape and is called sister chromatids. These sister chromatids are joined at a point called the centromere. In the prophase, the mitotic spindle begins to form. This mitotic spindle is responsible for separating the sister chromatids into two cells. Here is a picture of prophase. Here we can see the nucleolus disappears. Nucleolus is the covering of the nucleus that begins to disappear. Then the chromatin, that is thread-like structure inside the nucleus, condense into chromosomes. We can see the X-shaped structure, that is chromosomes. The centrosome separates. Then the mitotic spindle also formed. Next phase of mitosis is metaphase. Here also the chromosomes continue to condense. Then when we look through microscope, the metaphase can be seen as the arrangement of chromosomes at the equatorial plane of the spindle. Here in this picture you can see the chromosomes are arranged at the equatorial plane. Then next phase is anaphase. The centromeres of replicated chromosomes, that is sister chromatids, are split and the sister chromatids are pulled towards opposite poles of the cell. In the metaphase, they were at equatorial plane, but now the sister chromatids are moving to opposite poles. The fourth and final phase of mitosis is telophase. The picture here shows telophase. Here we can see new nuclear envelope forms around each nucleus. The chromosomes are unfolded back into chromatin. The nucleoli reappear. The cell continues to elongate. Here we can see microscopic view of different stages of cell cycle. The first one is interphase, then prophase, metaphase, anaphase, telophase, and the last is cytokinesis, that is the division of cytoplasm. Let us try to understand the stage cytokinesis. This is the second process which starts after karyokinesis, that is the division of nucleus, or we can say mitosis. Cytokinesis is a simple process. It have no phases involved. We know in karyokinesis there are 4 stages: prophase, metaphase, anaphase and telophase. But cytokinesis is a simple process. There is no phases. During cytokinesis, the cytoplasm of a single eukaryotic cell divides into 2 daughter cells. The cytoplasmic division begins sometimes during or after the late stage of nuclear division, that is telophase. During cytokinesis, the spindle apparatus partitions and transport duplicated chromatids into cytoplasm of the separating daughter cells. Then after telophase and cytokinesis, the daughter cell again enters into interphase. Here we can see a picture of cytokinesis. A cleavage furrow forms and it deepens. Thus the cell divides into 2. In plants, the cell division is little bit different from animals. Let us go through the differences. The animal cells contain 2 clusters of microtubules and centrioles. But in plant cells, this special structure, that is centriole, is absent. In animal cells, these microtubules and centrioles together termed as centrosomes. They are located at poles of the cell. During prophase, the microtubules gives rise to spindle. But in plants, there is no centrioles, but instead they have microtubules. These microtubules gives rise to spindle. These spindle help in splitting the cell during cytokinesis. Then in animals, the cell is split from outside by a contractile ring forming a cleavage furrow. We have seen that cleavage furrow in the picture. But in plants, there is no cleavage furrow. Instead, they have cell plate formation. A new cell wall forms inside the cell that grows outward until the formation of 2 new cells. In plant, the cell have cell walls. The cell wall is made up of cellulose and lignin. This picture shows the process of cell plate formation in plants. Like mitosis, there is another type of cell division called meiosis. This special type of cell division occurs in gamete cells. It results in the reduction of chromosome number. That is, the number of chromosomes are reduced to half. For example, in human there are 46 chromosomes in a cell, but in gametes the number of chromosomes is 23, that is half of 46. It involves 2 rounds of division and result in the formation of 4 cells. We know in mitosis 2 cells are formed. Then the significance of meiosis. It retains the specific chromosome number of each species. We know that new individual is formed by fusion of gametes. The chromosome number from male gamete will be 23 and that from female gamete will be 23. This together form a zygote with a chromosome number 46. That is, the chromosome number is retained. It also increases the genetic variability in the population of organisms because new individual is formed by the fusion of two gametes from two parents. Then our last topic is chromosome structure. We know at interphase the chromosomes are visible as chromatin fibers. During cell division, these chromatin fibers condense and chromosomes are visible. Each chromosome is made up of DNA and proteins. These proteins are called histones. Each chromosome contains hundreds and thousands of genes that code for several proteins. During cell division, we can see structure of chromosome. Here is a picture that shows the structure of chromosome. There is a centromere. The first picture is before duplication of the chromosome. We can see two arms. The small arm is called P arm and the other one is called Q arm. In the next picture, we can see chromosome with sister chromatids. The center part of the chromosome that hold the sister chromatid is called centromere. And the end part of chromosome is called telomere. In the last picture, we can see kinetochore. Kinetochore is a disc-shaped structure. From this, spindle microtubules binds. Here we can see a cell. The cell contains nucleus. Inside the nucleus, there is chromosome. When we unwind the chromosome, we can see histones and DNA. Once again, let us go through the processes we have studied. The cell division is of two types, mitosis and meiosis. Mitosis occur in somatic cells, whereas meiosis occur in reproductive cells. Mitosis can be divided into two parts, nuclear division and cytoplasmic division. Nuclear division or karyokinesis involves interphase and mitotic phase. Interphase passes through three stages, G1, S and G2, and it makes the chromosome material double. After interphase, there is mitotic phase. It passes through four stages, Prophase, Metaphase, Anaphase and Telophase. After nuclear division, cytoplasmic division or cytokinesis takes place. The cytokinesis takes place by a cleavage furrow in animal cell or cell plate formation as in plant cell. Chromosome has a centromere and arms. These chromosomes contain DNA and genes. This picture shows a stage of mitosis. Can you identify this? We can see the sister chromatids are going apart to poles. Let me know if you have any doubt on this topic. Let us meet again in another video. Thank you.